Okay, so good morning. I am Augusto, and today I'm going to talk a little bit about protein interaction networks, talking about topology, the eigenvalue spectrum, and also some models that help understand the mechanisms that of the evolution of protein interaction networks. So, a protein interaction network is a graph of all the proteins of an organism, and we represent each protein of the organism as a node in the network. And when there is an interaction between two proteins, we add a link between these two nodes, and we have the structure of a graph. And the mathematical background to study the protein interaction networks is the graph theory from math. And this is an example of a protein interaction networks for the Carnohabditis elegans, which is it's a worm, and it has about like 4,000 nodes. But to study it in this way is a little difficult because it's just a lot of dots and links. So we need different mechanisms to study and to figure out the properties and also uh, important uh, features and values of the network. So one approach is to study the topology of the network, which is basically the distribution of probability of the connectivity of each node, in which the connectivity of each node is the number of links that this node has. A network can be directed when you have the direction of links between two proteins matter. So if it's directed, a link from A to B is different from the link of B to A. And in an undirected network, the direction of the links doesn't matter. So the link from A to B is the same as B to A. So then we can estimate the probability of finding uh, nodes with a specific degree in the network. And then we can have the topology as a probability distribution function, or the PDF of the network, which is basically the probability of finding a node with a specific value of k, in which k is the degree of the node. So here we have the topology for 5 networks: C. elegans, it's a worm; Thaliana, it's a plant; Cerevisiae, bacteria; Bellinaso is the fruit fly, and also the network for Homo sapiens. And despite being a network from organisms of different uh, range and different origins in the environment, they all have almost the same topology, which is interesting because so we can have a universal approach to study these networks. And what we can see is that the topology follows almost a power law, and this is a log-log plot. So in a log-log plot, a power law would be just a straight line. And here we have almost a straight line, but we have this region with a high value of high connectivity and a small probability, which represents the hubs of networks. So it's not interesting to use the topology in this way because we don't have like a continuous function, for example. So we can represent it as using the complementary cumulative distribution function, in which we plot the probability of find the value of k higher than a specific value. So we have a more continuous distribution that provides a better information about the topology of the network. And you can see better that they all behave almost the same way, despite being from different organisms. Another important feature of the networks is the eigenvalue spectrum. Since the network is a graph, we can represent it as a matrix, which is called the adjacency matrix, in which each line, each row, and each column of the network represent one node of the network. So the elements of the matrix for unweighted network is just zeros and ones, and we add one in the element of the matrix in which we have a node. So if there is a node, if there is a link between nodes 1 and 2, the elements I1, 2 and I2, 1 of the matrix will be 1, and the others will be 0. So from the matrix we get the eigenvalues, and when we plot the eigenvalues of these matrices, we have almost the same shape for all organisms and some specific features, which is a high degeneracy at the 0 eigenvalue. And in a matrix, when we have a 0 eigenvalue, when you have 2 equal rows, and in a network, there will be 2 equal rows in the matrix when you have duplicated nodes. So 2 nodes with exactly the same link, with the same links. So it is an important feature because it gives a hint on some mechanisms that help that are behind the evolution of a network. So we have this high degeneracy at a 0, represent the duplication of nodes in a network. And we also have some degeneracy at the -1 and the 1 eigenvalues that are related to the presence of small subgraphs in the network. So we have small clusters in the network that are responsible for specific functions inside the organism. And we also have this characteristic shape. We have a small gap around the 0 eigenvalue, which is present in almost all the organisms that we are studying. Except for the cerevisiae, it's a little bit different, but we still have some minor gap. And to represent and to understand the evolutionary mechanisms of networks, there have been proposed some models. The first one is the Barabasi-Albert model. It was the first model that was proposed to reproduce and to understand how networks evolve, and it was proposed based on the network of the World Wide Web, the internet, and introduced two concepts. The concept of growing, in which we have at each time step there will be a new node will be added to the network, and the other one was the preferential attachment, in which in simple words, a new node will be added with more probability to the nodes with a high connectivity on the network, or the hubs of the network. And this model applies to the internet quite well.
2 minutes.
Okay. Because, for example, if you create a new website, you will preferably link it to Google or to other search mechanisms that are hubs in a network, so your website can have a high visibility. And it is characterized by the topology of the networks of the Barabasi-Albert model follows a power law, so we have a straight line in the topology. And for a long time, they applied this model for protein interaction networks, but it doesn't capture quite well the topology of the network. The second model is the duplication divergence model, which was proposed specifically for protein interaction networks. And it is based on random duplication of nodes. And we start with a small network, so you randomly select one node of a network. You will duplicate this node, so it will have exactly the same links of the parent node. And we have a probability of having a link between the duplicated node and the parent node, and also a probability of deleting or to represent mutations. So we will delete one of the nodes linked to the parent or the new node. And I'll show some results of this model later. And the later model is the hybrid model, which we propose as a different approach to represent both the topology and the spectrum of the networks, because Barabasi and Albert model and the duplication aversion, they don't represent both of these features of the network. So we propose this model as a combination of these two models with small adjustments to represent both the topology and also the eigenvalue spectrum of the network. And okay, and here we have some results. This is for the Ciel is the topology network. The first image is from the duplication divergence model. The black dots are the topology of the experimental networks. This red line is a fitting function, which is a power law with exponential cutoff that represent the points. And the blue dots are the topology for the models generated by the duplication divergence model, which is the error bars for this is like for 10 realizations of the network. And on the other image is the topology for the network generated by the hybrid model that we are proposing. And it has although we need some adjustment, but it has a better adjustment compared to the duplication divergence in the topology. And for the eigenvalue spectrum, this is the experimental one. From the duplication divergence model, it has the degeneracies expected from the network, but it doesn't have the gap and this characteristic shape. And from the hybrid model, we can reproduce almost the same topology, the same spectrum as the experimental network. And here's for another organism, the melanogaster. So the black dot's still the experimental network, red line the fitting function. And here the duplication divergence is a little better in some aspects than hybrid model, but when you go to the eigenvalue spectra, the hybrid model captures the specific features of the network. And to finish, here also for the Homo sapiens, the same results. We have a good agreement for the hybrid model, and in this case, the fitting functions not quite good for the power law exponential cutoff, but the eigenvalue spectra is almost the same as the experimental one for the proposed model. So it's the end. Thank you for.
Thank you. Perguntas, questions?